Oh, hold on. I'll just press the buttons I have to on the old socials.
And I'm going to mute everybody. No, I can't see where it's up here.
There we are. Right, okay. Feel free to go for it then.
Right, are we all muted? I'm still open,
No.
but there we go. That's fine.
Lost the button.
Okay, we'll make a start now. And good evening, and welcome to tonight's lecture at the North of England Institute of Mining and Mechanical Engineers. Without delaying too much, we'll conduct our short procedural agenda, beginning with apologies for members' absence. Does anyone have any members' apologies? I know I have Rick Smith.
No, I don't have any apologies at the moment, Steve. Shall I go on to our last meeting?
Yes, please do.
So actually, this was 21st of February, I believe. It was given by Dr. Hannah Hughes of Camborne School of Mines. We had a very good attendance. And since then, it's been very popular on YouTube as well. We had some excellent questions from the audience, especially one from John Theobald, who highlighted a few issues that had been occurring down in Balby back in the day to do with gas explosions along the shale beds. And I believe Hannah and John are discussing this further, so hopefully that will be an excellent research output for us there. The vote of thanks was given by Norman Jackson and closing remarks by the President. Any other comments, Steve?
None at all.
No, we'll take that as a record of the meeting in that case.
Indeed.
So our next lecture, we have a special event in April, but our next normal NEMI lecture will take place on Thursday, the 20th of May, and this will be by Professor Stephanie Glendinning of Newcastle University, and it's the title's to be confirmed, but it's going to be on long linear geotechnical assets and how you can monitor them due to the increasing complexity of the infrastructure we have. But our big event for next month is a 2-day conference joint with the Durham Energy Institute, and this is to do with the Northern Energy Transition. So we'll have day one is predominantly sessions that have been organised by our institute. We're going to go through the geological record of climate and extinctions. We're going to have a session on the geology for the energy transition, predominantly coal mine water heat down in places like County Durham, but also to do with critical metals as well. We'll also have a session on materials for the Northern Energy Transition and what local companies are doing and manufacturing in terms of advanced materials in our region. And finally, to end the day, we'll have a session on COP26 organised by the Durham Energy Institute of potential outputs from the conference. On day two, we'll have some representatives from local government, a 3-minute thesis competition given by PhD students from across the region, and also a keynote speech by Anne-Marie Trevelyan MP from Business, Energy and Industrial Strategy. And I'm sure many of our members will have a number of questions for her, and we'll round off the conference with clean energy and manufacturing solutions, including steel and perhaps what low carbon transport could do for the region. This is a public event. We'd love to see as many of our members and as many members of the public come along, actually listen to some science and hear what might be going on. And you also have an opportunity to ask questions as well. So do make sure you look it up. It'll be on our social media and also our websites, and make sure you register via Eventbrite.
Is this a live event or a Zoom event?
Yeah, just a moment, David. Yes, it'll be live. We'll have some questions at the end. So yes, don't forget to follow us online if you want to find out more information about all our events. We're at Twitter, Facebook. We'll have a increasing back catalogue of lectures on our YouTube channel, and also don't forget our website as well. And don't forget, if you're interested in hearing more about our events or joining in with our Zoom events, don't forget to go to mininginstitute.org.uk/membership and sign up. We've got a range of lectures and conferences, an annual dinner with men and women of professions, range of professions. We're working on some CPD, and we also usually in normal times hold field trips as well. So don't forget, we've got plenty of excellent benefits of membership, so please do go and look that up. Yes, so just to answer your question, David, yes, it's a live event, and we'll be sending out the Eventbrite sign-up link shortly. So you'll be able to ask questions on the day and submit them beforehand as well, I believe. So without further ado, I'll hand back to our president, and who will do the introduction for Professor Martyn Dade-Robertson. Thank you very much.
Thank you very much indeed, Andrew. Well, we have great pleasure in introducing tonight's lecture by Professor Martyn Dade-Robertson, who is co-director of Hub for Biotechnology in the Built Environment with Northumbria University. Martyn specialised in design computation with a special interest in synthetic biology, and firstly studied architectural design at Newcastle University, followed by an MPhil and a PhD at Cambridge. His early career was specialising in information architecture, comparing physical and digital information spaces, where he was nominated for the Royal Institute of British Architects Research Prize. Developing an interest in synthetic biology, heralded as one of the 8 great technologies, he recognised the gap between scientific state-of-the-art and design speculation in biotechnology, and became interested in bridging this gap. After completing an MSc in synthetic biology, he built a research group taking a human-centered approach to synthetic biology, his work receiving widespread media coverage, and is editor for the Routledge book series, Biodesign, and also over 50 peer-reviewed publications. I'm not going to say any more, it's taking too long. So now may I hand you over to Martyn Dade-Robertson to give us this very interesting lecture. Thank you very much.
Thank you very much. I'm going to see if my screen share will work. This is always a moment of trepidation with these things. It's working, but not the screen that I want you to see. So let me try that one again. So let me take you to there.
That's it.
And you can see that, and I will go into full screen mode, and then hopefully you can see that, and I will still be able to see you as well, if this works well. Okay, can you see that? Is that popping up on the screen okay, and you can hear me just about all right? Well, thank you so much for the invitation, and my talk today is going to be reasonably informal, so if people want to ask questions as I go through, I'm happy to take them. If not, then I'll be also happy to take things at the end. But you gave the introduction so well, I don't really need to do this slide, which is very helpful. So I'll skip back there. Yes, all of the things that you've heard are roughly speaking true. So I have this dual background, an odd background, I guess, in architecture and computing, and then synthetic biology, which is a field that you may or may not be aware of, but we will talk about that as I go through the implications of that. I'm here representing, I'm sorry for the low resolution slide, but I'm here representing the Hub for Biotechnology in the Built Environment. So we received funding just under 2 years ago from Research England to set up a new research centre across Newcastle and Northumbria University. And I'm particularly proud because it's one of the very few examples of a really proper collaboration around a single research theme across those 2 institutions. We should work together much more. We set it up with the idea that there's an emerging field of biotechnology that's coming along in all kinds of aspects of life, medical technologies through to chemical technologies and so on. But there are huge challenges in the built environment that could potentially be solved with novel biotechnology. So we got together, so myself leading for Newcastle and colleagues over at Northumbria University who were involved in biotech, put together a joint bid. The short of it was we won £8 million. And since then we've been building a new facility. We went from 6 people, we're now up to 50 people at this point, many of whom I've just been on a Zoom call with. And we're split into 4 main research themes at the moment. So building metabolisms, living construction, microbial environments, and responsive interactions. I'd like to talk about all of them, but I'm going to focus just on one of those themes today, given given time constraint. The overall motto of the hub is for the development of future built environments, which are life-sustaining and sustained by life, with an emphasis on that last bit, the sustained by life bit. That's what makes us unique. That's what makes us the research that we're doing particularly interesting, I would say. And I lead the living construction research theme. So I'm going to talk through that a little bit today and give you a glimpse, and very short glimpse really, into just a few of the projects that that we work on in that theme. Before going any further, I'd like to say that this is a huge collaborative effort. So I have a large research group at this point. Some of the names of those research group are on the board right now. These are some of the work I'll be referring, the students and researchers whose work I'll be referring to most of all, and my collaborators as well. So I collaborate across a range of different fields. I have civil engineers, biologists, microbiologists, synthetic biologists, architects and designers of different sorts. And our research is, I'm pleased to say, really well funded at the moment. So we have a number of different projects in different research funded streams. And I'm very unusual. I teach architecture. I teach how to design buildings, but I have a category 1 lab that I run. So it's not completely unique in the world, certainly very rare to find this kind of facility in that kind of context. We are setting up at the moment. You'll note the lab is quite empty. That's because we can't get back in very easily right now, but we're getting there. So the lab is ready. It was also a big day for us today because we're building an experimental house. And I'm aware I don't have a slide on this, and I will for future, but we're building an experimental house called the Ohm on the Newcastle University campus. This is going to sit just behind the Great North Museum. The steel work went in the last 2 days. I have to say steel would not be our first choice of sustainable materials, but it has to be steel for lots of different reasons. And it's going to be a really unusual and interesting building that we hope to open up in May, and will become also partly public as well over the next year or so. So there are lots of exciting things happening. So just to place this work in a little bit of context for you, and I'm aware I'm speaking to quite a diverse audience here, and so some of you may be aware of this kind of research, some of you may not be, but I'll just place it in the context for you. So probably one of the key emerging areas in architecture and building design is digital fabrication. And it's whilst it's not mainstream in construction yet, there is the thought that digital fabrication will become much more prevalent in the future of construction. I'm showing two buildings here, quite contrasting but similar in many ways, of buildings that come about as a result of the coupling of hardware and software, computer hardware and computer software. So the top building, Bilbao, is enabled because those forms can be made, because the drawings can be made and then translated directly into CAD/CAM using a process very similar to the way that they design and engineer jet aircraft, or any kind of aircraft actually, to create these bespoke forms. That's not a cheap building by any means, but it's a building that wouldn't have been possible 30 years ago, for example. And it was a pioneer of its type, developing this direct link between hardware and software, the manufacturing of the building components and the software they used to create them. The image on the bottom left is a bridge that's been constructed in the last couple of years in China, in Shanghai, from a group at Tsinghua University. It's a 3D printed bridge, so it's using concrete which has been 3D printed, extruded out, and then assembled on site. Those members are completely load bearing, so as I understand it, there's no specific reinforcement in there. It's been designed specifically to be 3D printed and then rolled out in that way. And 3D printing of buildings, so using the technology that we've seen on the desktop, allow us to make things, and extending them to the built environment is really prevalent. One of the interesting things about digital fabrication in design, architectural design, is that there's also quite often an implicit link to biology. This happens in two ways. So people are interested in biomimicry. So the top image is one of the pavilions, a yearly pavilion from Stuttgart University, a group led by Achim Menges. And every year they take an inspiration from nature, from a structure, for example, beetle shell, looking at how the materials are structured and then translating those into a digital fabrication method and fabricating them on site. In this case, you see the robotic arm behind there, stringing up carbon fiber, kind of a carbon fiber sewing process, almost a winding process to over a frame. And the other area is biointegration. So the image on the bottom left is by an MIT-based academic called Neri Oxman. It's called the Silk Pavilion. So it consists of an aluminium frame, which has been wound with silk, and then has been seeded with 1,000 silkworms. So effectively what the silkworms do is then complete the structure. So they're making kind of secondary structure by crawling over this structure and then filling in the canopy effectively. So it's a concept piece for how you can integrate a biological fabrication process through the silkworms with a digital fabrication process, in this case, robotic manufacture. So in my group, we see biology slightly differently to those other people. So we see biology as potentially wetware. So we describe it as wetware in the same way that we have hardware and software. And as wetware, biology, and so whereas these other fields have emerging technologies, enabling technologies, so typically the relationship between software and hardware comes about through software like CAD/CAM and parametric design, material-based design computation, where you can do modeling and form generation in software, and then translate that directly into, for example, the machine path of a robot or a large-scale 3D printing process. In wetware, we have the same principles. So we have enabling technologies, and specifically synthetic biology is my interest, and more specifically microbial synthetic biology. So synthetic biology uses a range of different techniques, including things like genetic engineering and systems biology, computational methods for biology as well, to essentially engineer microorganisms to be agents of fabrication, to fabricate materials. I'm going to talk about that in more detail. So we're very focused on the wetware bit, and our general question is a big one. So the question I asked my group, I would ask them every day, if I saw them every day, is can we grow a building? So can we go from constructing a building to growing a building? Imagine popping a seed in the ground, and your building then grows, self-assembles, organizes itself, use the resources available to it, to turn those resources into energy, to turn that energy into matter and produce a building. So that's a very big aim. That's a science fiction aim, but that's something that governs a lot of our thinking and our research. So I guess more specifically, I would say that in the built environment, we tend to use materials. After those materials, we do use biological materials a lot. So we use timber, we use limestone. These are the results of biological fabrication processes, but we use those materials after the organism that created them has died. So if we can start by making materials using the capacity of biological systems to grow, then we have a range of materials and construction processes that have never been available to us before. So we start with wetware. We take what we call a biologically first approach, and we mainly focus in two types of organism: microbes and fungi, and I'm going to go through those in a little bit. So when we think about these fabrication processes, we think about biology make things in different ways. And we think about 4 categories, or actually, I suppose 3 categories of the way that biology make things, and a 4th category, which is how biology might help us make things. So when biological systems make a material, and think of yourself in this regard, because you are, after all, a biological system making a material. You're made of a material. We've made many different materials. We think of the materials that make up biological systems as either constituted. So these are materials that are made of living cells. So most of you are made of living cells, for example. So your tissues are formed from cells. Those cells are part of a fabrication process that makes you. They are produced by living cells. So there's some materials that are made by living cells, but ultimately they're exported outside those cells and have a life of their own. So they become what we call an extracellular matrix. Or materials are induced by living cells. So they are formed through some change in the environment caused by the cell. And usually that's a chemical change that will then make the material self-assemble. And then finally, materials that are made active by the inclusion of a biological agent, by the inclusion of a cell. So this is not necessarily a fabrication process, but is the way that we could use cells within our own materials. So for each of these, I'm going to give you an example of projects that we're working on in each of these sections and how we're trying to make them relevant to the built environment. And a lot of this is very early work, but I want to try and project to you potential future. So starting with constituted, we work with a material based on mycelium. So if you've not come across mycelium before, mycelium is the root network of fungus. So if you've eaten the mushroom bit, most of the organism lives underground usually, or within whatever substrate it happens to be growing in this very dense and complex network of roots. That can often go on for miles, actually, in a forest, for example. If you see the mushrooms popping up, chances are there's a really dense network of this fibrous material underneath. It's really interesting as a material because you can cultivate it. And in fact, we're just talking to a mushroom farmer about how we use the waste from their mushroom growing, because it grows so densely that you actually get quite a decent bulk material at the end of it. So even a bulk material that has some compressive strength, so you can make compressive materials with it. It's also a really good insulator. So it's being actually developed by a number of startups as a building insulator at the moment. The beauty is it grows very fast, and it grows at room temperatures and pressures, and it will grow and bind whatever it grows on, and we can grow it on waste materials. So typically we grow it on things like wood chips or sawdust. We also grow it on coffee grounds. So we went through a period when the coffee shops were open of going around Costa and Starbucks. I don't know whether this is apocryphal, but apparently our mycelium prefers Costa coffee rather than Starbucks coffee, but I don't know whether that's the case or not. We can grow it within a few days into quite thick mass and material that when we dry out and use. The image on the right hand side is not our work, actually. This is a company based in New York called The Living, who used this process to make these bricks. So these bricks were grown, then they were compressed. So they were actually compressed in order to give them a little bit of strength. And then this pavilion structure was built with them to show that they could be used in this way, which is really fascinating. So in our group, we start with a principle that we took from Louis Kahn. So the famous architect Louis Kahn is often quoted because he also taught architecture as well as whispering. He's a brick whisperer, we might call him. So he would say to his students, if you're out of inspiration, ask your material what it wants to be. And he started the dialogue, you ask a brick, what do you want to be, brick? And then the brick will say, I want to be an arch. And you carry on the dialogue till you have a building. So in a similar vein, we say to mycelium, do you want to be a brick? And our principle behind that is that mycelium is a really complex biological material, and it has lots of potential that we might not exploit if we think about it in traditional building terms. So this is the work of one of my PhD students, Dylan Oscar, who's built these, what she calls robotic growth chambers. So these are growth chambers where we're growing on mycelium composites, but we can quite carefully control the humidity, the temperature, the airflow, and other things. And what we see, and that's what you're seeing in the bottom image, is that for different conditions, the mycelial network will grow into different forms, into different morphologies. The important thing there is that the different forms of the mycelial network within a material lead you to different material properties. So with mycelium, you can get from a material which is quite rigid to one which is actually quite flexible, maybe be cushioned. Some materials are quite friable, so they're easy to break apart. Others are fairly well formed. And so we can then map those morphologies to these different properties. So you don't just get one material, you get a whole library of different materials from similar techniques, dependent upon the environment in which you grow them and also the substrate that you use to grow them on. And what's really interesting as well is that mycelium will eat some of the substrate. So they'll use that as food, but they'll also act as a binder. So treating the substrate and an aggregate material effectively. And then in other times they'll fuse to the material as well. So they'll actually fuse these aggregates together. They'll form parts of the aggregate, but where they're not necessarily distinguishable from the aggregate, but you've got two different materials going on there. And then this is a work of one of my undergraduate students who took that idea and made a chair using this casting technique. So what you're seeing there is actually modeled on an IKEA chair. In fact, we used an IKEA chair as the mold. So this is one of these plastic molded chairs you can get from IKEA, and we used that as the mold, or at least Natalia did. She then packs it, in this case with a combination of wood chips and the coffee grounds that I was talking about before, keeps it moist. Seeds the mycelium then grows, and she leaves that for 2 or 3 weeks. Then she lifts the material out of the mold, dries it, and you've got a really solid chair, actually. So you can sit on that. It's perfectly stable. What's really interesting is that it's while it's a monolithic casting, so it's done in one go, the outcome has different material properties to it, or has the potential, and it didn't quite work fully in this case, but has the potential to have different material properties. So you imagine the chair that you're sitting on, it's probably made of more than one component. You've used different, the designers have used different materials to give you different strengths. So the back might offer some cushioning and some flexibility. The legs offer rigidity. You can do that within one cast. So you can have a single cast where you have those different functional properties there. So that's really exciting starting point. So to take that to the next level, we think, well, can we use this as a building method? And believe it or not, our biggest client for this kind of way of thinking is NASA. So we're working on a project with NASA, and Monica Lipinska, who's just started a PhD with me, but also who worked for NASA before coming to work with me, who are interested in using this technique as a way of building habitats on other planets. So that might sound kind of crazy, but that's what NASA think about. And they're thinking about scenarios where you can't just go and take a cement mixer with you and a bunch of concrete, like Mars, for example, but where you may well take the seeds of mycelium and then use the biomass created by human habitation to begin to develop and expand a colony, and use this idea of functionally grading the material to adapt to different conditions. So you may not use this as your only building material. In fact, you wouldn't use this as your only building material, but imagine a building where the walls could also be used to harvest food, for example. And we're also working with mycelium species that offer things like UV protection because they contain melanin, other useful protection against those things as well. So she started working on some concept designs using this approach, and then we're deriving experiments that break down this approach into different parts and see just how big we can construct using these methods. So that's kind of an exciting project, and we're very excited to start then. And NASA have just applied for some funding through their scheme to take that to the next level. So now going to the next category, the produced materials. And so the one that we're most interested in is known as bacterial cellulose. So cellulose is a material that you'll be very familiar with already because it's the basis of wood. So wood being a cellulose lignin hemicellulose composite. Bacterial cellulose is kind of the same stuff, but it's a much purer form. It's grown in interest a lot recently, particularly the design community, because it can be seen as a replacement for plastics and leathers. So it can be manufactured in different ways that make it a much more biodegradable alternative to plastic, for example. There are lots of different ways, lots of different applications for this. So there are beauty treatments, and it's used as wound dressing in its hydrated form, and then in its dehydrated form. So once we've dried it out, we use it. It's been used in various products, including like high-end music speakers have been using it as a kind of membrane material. And designers have been interested. There've been people making jackets and other things with it. It has some real limitations in that area, I have to say, but it's of interest. Architecturally less so. It's quite hard to scale up production. And it's also quite, initially, it's most of the material is quite homogeneous. So it's most of the applications you see use fundamentally the same process for manufacture. So we want to go beyond those ways and ultimately scale up. And so we applied for and did run a project that started at the beginning of March to develop a new fabrication technology based on growing this material. So what you're seeing on your screen, and I'm sorry this video will likely be very stuttery for you, this is the nature of Zoom, but is at the heart of this setup is a bioreactor. Bioreactor, just a big chamber that we feed a liquid culture which contains bacteria in a food. And that's hooked up to a computer and a projector. So the projector is able to project onto the top surface of this bioreactor. So you see the food being passed in, and running on our computer is a 3D model. So we're going to take a zoom into the bioreactor, and you see two types of bacteria there now. So the green rod-shaped things are our cellulose-producing bacteria, and they work by floating on the top of the liquid culture and then producing this dense network of fibers. And they do it very quickly. It appears initially like a skin on the top of the material, and then will thicken up over time. And we believe we can grow it to quite some thickness. And then we have our little white bacteria in there. They're E. coli, and we've engineered, or we will have engineered those to be light sensing. So they're able to respond to light by producing a pigment. And then we're projecting light onto the surface of the material. So where we project that light, pigment will form. We then grow the cellulose culture. So it grows, and then we change the pattern of light corresponding to the section through our model. So we're now building up this, this section. The idea is at the end of it, we'll have the pigment in 3 dimensions within this pellicle. So we'll dry it, it will shrink, we will open it up, and we will see inside. So that doesn't sound super interesting. I mean, it is really interesting for us, but just being able to pigment a cellulose pellicle is not necessarily a terribly useful thing to be able to do. But it's a proof of concept, really, because if we can do it, what we can do is to co-culture our bacteria with other types of bacteria that produce different sorts of material. So the image I am showing on the right, that kind of diagram. So imagine something a little bit like a printer, but rather than getting printer cartridges, you buy a vase of bacteria. And you might have a bacteria, for example, that biomineralizes. So it might leave mineral crystals. We'll have programmed it to respond to those light patterns. So as we shine light, it will produce mineral crystals where you want to. Or an enzyme, for example, will actually break down the cellulose as you're growing the cellulose up. So an enzyme like cellulase. So we can actually mold 3D prints as we're going along. So we create very new sorts of materials that could be functionally graded and could be made to be quite complex for a whole range of different applications. And ultimately, we'd like to scale that process up for building. So for example, building membranes that we might design and actually grow in situ on site, almost like tissue engineering, but a very large scale to provide canopies and other sorts of structure. And my PhD student, Sunbin Lee, is working on this scale up process, imagining how we might get from the core science that we're doing in the lab to something that will work at a much larger scale. And this is a really exciting project for us, and we're lucky we've got 2 years worth of funding to do that proof of concept. So that's the produced materials. And so the third in my list is the induced materials. And for that, we're interested in the process of biomineralization. So biomineralization is another biological fabrication strategy that's used a lot in nature. Your bones, calcium phosphate, your bones and teeth, calcium phosphate is caused by this process. Shells, of course, an example of calcium carbonate and the control of calcium carbonate to make structural form. The one we're interested in is called MICCP, or microbially induced calcium carbonate precipitation. It's already used to some extent in the building industry. Henk on the right hand side there, I'm pleased I've just been in a meeting with Henk. Henk was chairing our steering group today, but Henk has worked on a self-healing concrete using microbial spores. So what happens is that these microbes create an enzyme that ultimately, and I won't go into the chemical basis of this, but changes the pH of the environment. In a calcium-rich environment, that leads calcium carbonate crystals to form. And what Henk has done is to develop a type of material that's self-healing. So there are little, within the aggregate of the concrete, there are these little spheres of a food and bacteria spores. They're like seeds for bacteria. When the material gets cracked, and water gets in there, the bacteria come alive, they trigger the process of biomineralization, and the cracks will reseal again. And so it's been proven now on real civil engineering tests that this works and makes the material incredibly robust and able to self-repair. And Henk is setting up a company working on this material now. The one below is Ginger Dossier, who's created a company called Biomason, which uses the same approach, but this time to actually create an alternative cement. So she mixes the bacteria with an aggregate and the appropriate food stuff, and they cement a solid material at the end of it, in this case creating bricks. They do so with low energy input. With low energy input, and in a potentially a carbon negative way, because they take carbon dioxide out of the atmosphere in that process. It's not a super scalable process yet, so we won't see it replacing concrete anytime soon, but it's certainly an idea that this way of making materials might become more prevalent. So the question in our group is how we could make this MICCP process more intelligent. So could we make a material that's able to remodel itself in response to mechanical change in its environment? The influence for this is really the way that bone remodels itself. So if you are a marathon runner, for example, then the bone density in your shins is going to be greater because the cells that make your bone are responding to the repeated mechanical loads by synthesizing more bone material to give more strength in your bones. So that's our thinking. Can we connect this microbial process to create biocements that will respond to loads? So you have a material, you load it, and where you load it, it reinforces itself effectively. It makes more density. So this is a long project. So when I first got into synthetic biology, this is the project that I started in the lab initially, and then we've developed as funded projects since. So what you're seeing on the left-hand side are two images of bacteria. So those little rod things, the green rod things are the bacteria, and they're producing a protein known as green fluorescent protein, GFP, which makes them glow. So think of it like a light that you can turn on. It makes them glow under UV light. What we've done is to search out for genetically sensing, pressure sensing genes in bacteria, and we discovered that nobody had looked into these before. We actually found quite a number in our species bacteria. And then what we did is to engineer the bacteria so that as we put them under load, they light up. And that shows that our genetics, we call a genetic switch, is able to take input and then give us an output. And then the image below shows, again, same sort of bacteria. You see the little rod-shaped bacteria, but this time they're synthesizing calcium carbonate crystals. So that roughness you see on the surface are calcium carbonate crystals. So as the material, and what we've been able to do is to start designing genetic circuits within the bacteria. That means we can turn that process off and on when we want to. So the idea is we connect them together. So as we load them, the process is turned on, we get cement. If we're not loading them, we don't get anything at all. So we have those two systems working independently now. And what we're doing in the next 6 months, hopefully, is to put those systems together and make a single system. A microbe, bearing in mind these are 2 nanometers across, but within a material we can have trillions of them. Sorry, 2 micrometers across, not 2 nanometers, 2 micrometers across, but we can have trillions of them, all sensing their environment and responding. We also then try and scale up. So this is the work of my PhD student, Thora, who has been looking at using this biocementation process to make larger casts. And we're very excited at the moment because we're just taking part of the process, but we're hoping that we might be able to get some funding to work with a local company called Bespoke Concrete, who are really interested in using this as a casting method for making decorative castings and other things. So the process is deceptively simple, really. We have a sand aggregate. We then we set that within a mold, and then we pump through the mold a combination of we seed the sand with bacteria, we pump through a nutrient and a reactant containing calcium, and then over the course of about, well, in this case, it's usually a course of about a week to 2 weeks, we then get a cemented cast left over. These things are about the size of bricks. We think we can go a lot bigger, so we want to be able to cast quite large volumes of material in this way. But it's quite a complicated process because you're dealing with a living cement. So you get unevenness, you need to think about how you have flow of the liquids through the material. And so we've done a lot of work developing these novel casting methods that we're hoping to be able to scale up in the next year or so. The other aspect of this is to develop new computational methods. So we're trying to connect the kind of modeling that we would do in, say, civil engineering. So looking, for example, at finite element analysis to look at forces through a material and connect that to how we know the material will behave if it contains our bacteria producing the enzymes that make our material. And so this involves quite a lot of novel computation work as well. And we've been developing some new versions of new tools for visualization. So our aim in the first instance will be to do something like the top right image. So the top right is an odometer. That yellow stuff is a jelly containing a bacteria, and we're loading it. And the idea is that as we load it, it will begin to calcify. So as we load it, it will resist the load by calcifying itself and becoming stronger. So that's the demonstrator, and we hope to get to that demonstrator this year. that would be quite a major breakthrough, because that's a very new sort of material. A biologically active material in that way has not been demonstrated before. And we're projecting into a future where this might have applications for example, civil engineering. So one of our most far-reaching imagining of this would be the idea of creating a self-constructing building foundation. So rather than digging a trench, filling it with reinforced concrete, you'd seed the ground with our bacteria, and the soil itself would respond to the loads placed on it by cementing itself and creating a self-constructed raft. So this is just to say we've published quite extensively in this area. So this is the project that I've published more on than anything else. A lot of it's still concept work. We've got a lot of individual bits of science going on at the moment. We're just getting to the point now where we can pull them together to make our demonstration, and we're really excited by that. So my final classification of materials are known as hygromorphs. So these are, just to recap, these are active materials. So they're materials where we use a living cell to give our material an additional functionality. So we're interested in these hygromorphic materials. So materials are hygromorphic if they respond to water by changing their shape. So if you live in a reasonably old house with wooden doors that have warped and don't fit anymore, the chances are that's because the wood has absorbed water, they've changed their shape, and then they no longer fit into their frame. So usually this idea of wood warping is something we don't like, we don't want to happen. But some enterprising academics, again, University of Stuttgart, the same group I referred to earlier, has developed materials that use this warping to an interesting effect. So the image you see on the top right is the hygromorphic pavilion, it's called, or hygroskin pavilion. So there's little flaps that you see opening and closing are actually wood composites. So they're very thin sheets of wood that are then, which are an active material. So these will expand in contact with water, and they've applied them to a substrate of that is not hygromorphic, so it won't expand towards. In this case, it's probably a resin or plastic backing. And then what happens is that as one layer expands, so if it gets damp, for example, if it rains, one layer will expand and push the other layer. So you get differential expansion and you get this change. And that can be used to open and close flaps. So it's essentially, it's an actuator and a sensor all in one. And it's been demonstrated actually, and we've had some experiments running in Newcastle. My colleague Ben Bridgens and Graham Farmer have worked on versions of this material at work, and have been shown to be quite robust. So we've left them outside for a year and seen them in all kinds of different weather conditions and monitored them. And they can be made to be quite robust. But they're quite slow acting, and they're not super sensitive either, so they tend to respond to wetting and drying. We're working with a similar sort of material, but this one uses bacteria spores. So bacteria are the seed form for the of a bacteria. I mentioned those before. They're incredibly hygromorphic, so they'll expand and contract in response to water. They're tiny, but we can make solutions of trillions of them that we can then apply to a surface. So we've developed our methods based on some work that was done at Columbia University, the paper I'm referencing here, where we apply them to a surface of a latex. So we apply to the surface of the latex, and then we have these little mini actuators, and they're incredibly sensitive to slight changes in humidity, so they can adjust themselves in response to the humidity in the air. So our process here is to start by thinking about how we can program these materials by applying the spores onto the surface of the latex in different patterns. We can also program by building up different layers of spores. So we've worked out that we can, by adding layers of spores up, we can make the program respond more or less to these changes in humidity in the environment. We're also trying to then scale up and make them robust. I'll just show you this video. Again, it will be probably a little bit jerky for you guys, but this is sped up by about 4 times. So we've gone from a humid environment into a dry environment, and you just see it folding up. And then we can connect that a little bit like origami pieces, but where the pieces can start to fold up and open and close on their own, depending on the ambient humidity. The scale for this is about 2 centimeters for that one of those strips. So we can scale it to the next level. And then what we want to do, and what we've been working actually with our students in studio with, is to try to turn this into a building system or building systems, to think about how we could scale that up. And this is one of my favorite examples of a project for the past few years. Vincent designed the sort of interior membrane of a dance studio. So the idea being, as the dancer moves around and generates humidity and body heat and so on, the walls begin to flex and change and open up apertures and begin to move, which is a really interesting idea. And I think we're getting closer to being able to produce a prototype that begins to demonstrate that potential. And we could see it being used in the context of a building that might sweat. So another example idea that we've had is to put it in bathrooms and have tiles that begin to open up as you have a shower to allow ventilation through, and then close when you've finished. And we've got some cool ideas that we're hoping to apply in our experimental building in the next couple of years. So that was a whistle-stop tour through 4 material types. I could have talked at length about some of those and introduced more, but just to say that what we're wanting to do is to bring all of these things together. So that's the next step. So we want to be able to actually make systems where a lot of these different materials and technologies are brought together in a different way. Just to give you a little heads up on what we're working on at the moment. So this is a grant application I'm working on for bringing together these different manufacturing techniques into one place. So imagining a kind of factory environment where we can make material at scale in an integrated way, so we can make the material work in different. We can make materials in the factory setting that are functionally graded, that might make use of our cellulose, that might make use of our biomineralized materials and so on, and make building components from it. And we think the way to do that is to bring different microbes together and make new sorts of bioreactor and digital fabrication unit. We've also been dealing with new kind of materials and so on. So this is an example of a concept project that I'm working on called the Garden Bridge, which uses this as or the Gardened Bridge, I'm sorry, which uses this as a concept for making a structure. And that's just a sneak preview of something that I'm going to produce very shortly. So in answer to how we grow a building, the answer is obviously no. We may never get to that point, but the combination of these different approaches might enable us to make really interesting materials in interesting ways, and ways that are self-assembled and complex, and but done at very low energies in very new sorts of ways. I wasn't sure what my time limit is. I have an extra bit of the presentation that I can take you to a slightly different part of our research if you would like, or I can stop now if you would prefer. I'm not sure how people feel. Should we keep going? Okay, so I've got maybe another 5-10 minutes, but I just wanted to reflect a little bit on the current situation that we find ourselves in. Maybe talk about one of the other themes. So the other theme is the microbial environments theme. It's not the theme that I lead, but it's one that is increasingly occupying my attention. I think this might be of interest to everybody, really. And I think it doesn't matter whether you're interested in technology or not, this is quite important. So when we put in the bid, we set up the microbial environment theme because what we were aware of is that there's a growing literature on the relationship between the microbiome of our environment and human health. So there's something known as the hygiene hypothesis. And so the hygiene hypothesis is the idea that we've been cleaning far too much, and that children in particular brought up in very clean environments, particularly urban environments, tend to have higher instances of things like childhood allergies and so on. And the children brought up on farms or with lots of animals around them tend to do much better health-wise. And so we need to think more carefully about the microbial environment when we design our buildings. And we kind of threw in, and that might include viruses and other things that are quite important to the way that we use spaces. So that was 2 years ago. And then last year, we know everything that happened. So what happened was that as part of that theme, we bought all of this sequencing kit. So we have these very expensive devices that are able to sequence biological cells, so they can tell you what the DNA is in biological cells. Because we had one of the latest versions of this kit in the country, we very quickly became a node in what's known as the COG network. So when you hear that there's been a new variant of the disease, that there is this new mutation that's leading to bad health outcomes for people, it's because Our group, the group led by Darren Smith at Northumbria University, who's part of our hub, or somewhere in part of the COG network has found this new variant and is tracking it. So we sequence an awful lot of the positive cases for COVID-19. What this creates is a huge database of viral transmission. And so, and bear in mind, I'm not the expert in this area. What Darren and his colleagues tell me is that this level of granularity is so good that in some cases you can actually trace the path of the disease through individual buildings. So you can actually look at hospitals and see, for example, the transfer of patient to patient as they've gotten COVID-19. Because there are point mutations. So there are point mutations in every single new viral infection that give it essentially a distinctive family tree. We're generating huge amounts of this data, not only at Newcastle, but across the country. And this provides us with an incredible insight that we've never had before. And this is going to form the basis of research for years and years to come. The other thing that we've seen is the development of the rapid development of sequencing technology as well. So, if you've been, we've all come up through this kind of world of computers where I will tell my children that the same thing that we do on our mobile phone is the thing that used to take a whole room in the basement of a building. Well, sequencing technology is a little bit like that. So to sequence the genome of an individual bacteria, so 20 years ago, might have taken 5 major labs internationally, 10 years and multiple millions of pounds to multiple hundreds of million pounds to do. The thing that you see there is an Oxford Nanopore sequencer. So Oxford Nanopore, one of our project partners, that's the size and shape of a USB stick. It sticks into the side of your computer and it will do sequencing for you. Good enough to tell you what organisms you have living in your room. And this has been, these sequencers have been used to do a lot of the rapid testing that we've seen. So we're seeing a huge change in sequencing technology, and that sequencing technology is a bridge between the biological world and the physical world. So imagine, for example, a building that was able to detect a virus before anyone else was able to do it through a sensing system that was rather like a fire alarm for you, right? So these things are increasingly possible, or have the potential to be possible in the near future. So we've started to really rethink our relationship to biology through COVID-19. So what we've started to realize is that we're interconnected through this biological network of viruses, of microbes. We're also aware from our research that biological cells can sense things in the environment that are very hard to do using digital sensors. They're also carriers of information. So what is a viral network if not a social network? It's a map of social contacts between people. So well before we had Facebook, viruses were social networks, whether we like them or not. And they're also incredibly robust. So they don't need any input from us to survive other than the hosting in our bodies quite often, or in the environments that we create for them. And they're intimately connected to health and well-being. So we think a major direction for architecture, buildings and engineering in the future is going to be taking into account this biological environment that I think we've taken for granted for too long and we can't afford to do anymore. So something we're beginning to think of, and it's just an opening thought really, but is it to think about an internet of biological things, to think about how the sensing that we already do in the environment for things like pollution and traffic flow and all the domestic sensors that are around us, whether we like them or not, in our mobile phones and other things, also need to be connected into this biological world in new ways. And so we're thinking of future built environments that are biologically enabled. And we think this is really the frontier for the research that we're going to do over the next decade. So that was the extra little bit. I have a little bit on the end, just in case we have the time. Just to say that, I mean, I'll take questions now, of course, but we have a website for the hub, the hbbe.ac.uk, where we've got quite a lot on the different projects that we're working on. My old research group also is symbio.construction. We kept it very up to date up until last year, but it has quite a lot of information on some of the projects, and we've got some videos and things. I tend not to show very many videos in Zoom calls because they never work super well, but you can go and you actually see some of our stuff live and see people making things and see some of the work that we've done. And I'm always happy, of course, to take people sending me emails about random stuff. I've just had an email from someone at the BBC who works for a forensic science drama show about whether they can use biomineralization as part of a murder scene, which is fascinating. So I take all questions and try to give back the best answers that I can. So yeah, that's the end of my presentation. I can end my show there. Maybe stop my screen share as well.
Well, lovely. Thank you very much, Martyn. Fantastic. So do we have any questions? Yes, John, is it? Let's see.
Fascinating stuff. The materials are obviously developable. I made a word up. Are they also biodegradable?
So some are, yeah. I mean, so this is one of the things. Actually, the greater challenge that we have, John, is that sometimes they're a bit too biodegradable. So you kind of, if you're building a building out there, you don't want it to biodegrade too quickly. That's going to be an issue. The big sell of the cellulose-based materials over, for example, plastics, is that they are very biodegradable. So there are lots of enzymes that can break them down. The other thing about the materials, they're programmable. So there is the potential that we can add in agents that we can trigger using some very simple chemical process that will begin that biodegradation process down. The biomineralized stuff is harder to deal with. You know, mineralized materials generally are not necessarily biodegradable. So, but then we are looking to develop those for real longevity. So we're not interested in being thrown away materials, particularly. Interestingly, the mycelium materials are quite hard to deal with. One of the reasons why, so we were approached by a mushroom farmer who will plant the mushrooms in bags usually, and then he gets this real solid mass of stuff. And it's not like you can just kind of replow it in the way that you might, you know, for the remnants from carrots or whatnot, and you replow it into the ground, it just kind of rots back down, and you've got your the future compost. It's really hard to deal with. So we can take it away. If we can do something with that mass and make it into something, then he's really happy because he gets rid of his waste and we can make it into something. But yeah, all of these processes, the theme I didn't talk about is our building metabolisms theme, which is dealing with exactly that. So it's also biological processes that can break down things as well. And so a project I didn't talk about is that we're working with enzymes that can break plastic down, which is really exciting. So we're hopeful that, for example, I'm imagining the future compost bin where you like the super compost bin, where you don't just put all your food waste in it, you put your plastic bottles in it, and then after a couple of months it's all rotted down into smaller molecules and it's food for something else. Biologically that's possible, so that there are enzymes that will now break down plastic, and there's a center called the Enzyme Innovation Center that we're starting to work with that do this kind of work. So yeah, the idea is to see these as part of a whole material life cycle where the biology might make them, but the biology also might break them down.
Thanks, Martyn. Fascinating.
Excellent. Anyone else?
Yeah, can I just... that was really exciting, but also a bit terrifying as well, really. You know, the shades of Jurassic Park come to mind. You know, there's no danger of these things going off and doing their own thing, is there? You know, they building a living house that might decide to move, you know.
Yeah, well, not our living site. It's pretty heavy, to be honest. But no, it's a fair question. And so there's a simple and a complicated answer to the question, David. So there is a genuine, and I don't think completely unfounded, concern about genetic modification of anything. And you can call it the Jurassic Park fear, if you like. The systems that we work with are pretty straightforward and pretty simple, and the microbes that we work with are all pretty safe. So we work with a species called Bacillus subtilis, which you will, if you have a salad sandwich, you'll eat quite a lot of, whether you know it and like it or not. It's part of your life. We also work with an E. coli. Now, you'll have you'll come across E. coli in a very bad light because it's generally associated with food poisoning. But we use a type of bacteria, E. coli, a species of bacteria, which lives happily in your gut and is actually very useful to you. When you start to genetically modify things, I think there's a question about whether you bring genetically modified organisms into the environment and you let them loose.
Yeah.
Now, it's not like dinosaurs in that sense, but biology evolves and it changes, right? So you start with is not necessarily what you end with. And so quite a lot of our focus is on replacing processes that can happen within clean and safe environments. So genetically modified bacteria, for example, are used a lot in the chemical production industry, but they're used within environments where you keep them enclosed. They're in labs or factory settings where they're kept within bioreactors. And we also have safety measures, so you can add in what are sometimes called kill switches into the bacteria. So they have to be fed a very, very specific compound, or they're not able to survive outside the bioreactor. When we get to environmental applications, if you see my example of the living the kind of self-constructing building foundation, you would have to seed them in the soil. So they have to become part of the environment. And I think that's where we may find some barriers legislatively, ethically. We would have to think very carefully before we took that route. So I put it there as a provocation, but it's something that we think about quite a lot. And I should say within the hub, one of our priority hires was a social, in our case, a social anthropologist who's also has a background in areas like bioethics. So to think about the application of what we do and what really the limits of what we should and shouldn't do with our technology. So there's nothing really to be fearful of, I don't think, in what we're doing, but there are definitely ethical lines that I think we wouldn't want to cross, and broader conversations that I think society would want to have with itself before some of our technologies came on stream. So it's a fair question, and it's actually variations of that. One of the questions I do get asked quite a lot by people, particularly members of the public as well, when we're doing some demo stuff, who are very aware of those kind of the Frankenstein nature of what we might do, which is, yeah, definitely causes some concern.
Lovely. We've got a question from YouTube. How could growable buildings be prevented from rotting or being consumed? Maybe you've just
or indeed getting disease.
Yes, they get your house gets a virus.
Yeah, absolutely. I think there's a line I would draw that we've not yet crossed between growing the materials, but then effectively at the end of the fabrication process, the material is usually killed or dried, right? So the mycelium stuff, we dry it and we leave it. Now, it does have the potential to come back to life again. And an interesting problem is that, of course, mycelium is actually dry rot in buildings. So if you've got mycelium living in your house, you don't think, oh, great, my house is growing itself. You think, oh my God, the floorboards are going to break up. So there is that potential for life again. But generally, with the cellulose materials as well, we will dry them and treat them so that they are effectively inert by the time we finished. With the active materials, the spores that you see are spores, they're seeds for the bacteria. So they don't have metabolism, they don't grow. They retain the potential for life, but it's very unlikely in the conditions that we give them, they would be able to come back to life. And in fact, we can also tweak with their genetics as well to turn off that capacity to regerminate. So generally, the materials that we're using at the moment, there will come a point where we take them and they're effectively killed, or they're taken away. That doesn't mean to say that we don't have the ambition to use living materials, so materials that would retain life and would go on to live past that first construction phase, to do things like self-heal or grow, expand, a house that might grow itself. That would be an interesting idea. And in truth, I don't know what the answer to that question is. I mean, it would be a challenge to have a growing building and not to have it attacked by other species. So one of the things that we do a lot in the lab is that we take microbes, for example, that usually exist with lots of other microbes. And because of that, living in a community, they're very robust. And then we take them and refine them. We take one species and we cultivate those species particularly. You stick those back into the environment again, and they're not very happy because they're then attacked by other species and they don't have their for want of a better words, their mates around them to support them. So one of the principles that we're starting to think about is the development of multimicrobial systems. So where we give that kind of quorum benefit, so that the microbes are benefiting one another. So some of them are defending against enemies, others are metabolizing things that perhaps the other microbes can't metabolize. This is a really interesting area of research. It's very complicated to engineer, really complicated to engineer, but that leads to plenty of research papers, right? So we don't worry about that too much. But yeah, but I don't have a good question to that, and it is a good question.
Excellent. Is there anyone else? Is that David?
I've heard about them developing technology to go to Mars, to kind of send out before the humans go, to start building the resources that they can then use when they arrive. Are there any thoughts about how well this would work on another planet?
Yeah, so I mean, this is not my main area of expertise, and so we've talked with NASA about this, and there are various different scenarios that are put together in this regard. So one is that we'll send out robotic probes that will land much in the same way that the Mars lander that you saw, I've forgotten its name now, but landed recently. But rather than just simply retrieving samples, it will begin to start collecting together raw materials. NASA are working on an idea of essentially there's a multi-step process where you would send probes initially to begin to gather together raw materials. You then send a crew, a crew of astronauts who would land and essentially inhabit the vehicle that they land in, but then we'll begin to expand that vehicle by making use of local resources. And so the mycelial stuff that we're doing is part of that idea, that you might take the kind of seeds for the mycelial growth. If you've got enough that you can start create biomass, so you can start to grow things, then you grow things, you create biomass from that growth, you begin to then generate a material, great insulator or whatever, you begin to expand your habitat. So something that Monica is working on is the idea of a habitat that will begin to unfold over time. So you would begin to grow new bits of it. And it might use a combination of the biomineralization approaches as well to solidify. So I wrote a paper on this about how you might use biomineralization to make essentially as a cement in a Mars-like environment. There are a lot of unsolved problems to think about. Like, it's hard enough thinking about how you're going to keep humans alive without all of these biological systems as well. It's super cold as well, but in some ways that's a benefit. So if you can create mycelial materials and then freeze them, and you've got something that's quite strong as a structural material, for example. So there are payoffs, and there are lots of different concepts about how to do this. And NASA are investing in a number of different versions, some of which involve biotechnology, some of which involve other things. So they're spreading their bets a little bit.
Lovely. Anyone else? No. Well then, Steve.
There we are. Yes, absolutely wonderful. Well, it leaves it to me to call upon you, Andrew, to give a vote of thanks here.
Well, yes, thank you very much for that, Martyn. It really was an absolutely fascinating overview of what looks like a very sort of fruitful, complex yet rewarding topic, I'd say, with plenty of real-world applications. Sort of the way you went through the 4 different types, I particularly liked. Well, yes, I mean, whether or not you're going to rival IKEA would certainly be interesting, and I particularly liked sort of asking your mycelium, would you like to be a brick, or what would you like to be? It's always good to provide opportunities to see what things we'd like to turn out into. Certainly
going to IKEA though, and going into shopping, growing your own. That would be the thing when you get a bag of seeds in their mold. I would give away my Allen keys for that, I can tell you.
Superb. I also very much like these what you said about the cellulose, where it's less 3D printing and more 3D growth. Again, so see what you can do with your IKEA. Certainly, coming from a geological perspective, biomineralization is always of interest. And recently, our institute had an inquiry of how, if anyone had any novel ways of dealing with concrete rot in civil engineering assets. And so it's the potential applications there would be huge if that could be made to work. And as well as the hygromorphic materials and those different applications with their differential expansion properties as well. And then also as well, the range of future work, what sort of applications and sort of factory methods you might need, but also this biological Internet of Things, where we're looking at a really interesting way of sort of, how you can take a range of complex interrelated systems and give them to human benefit in their built environment. So on behalf of the Institute, thank you very much, and we certainly look forward to seeing you in person in the future.
Yeah, I would love to see you all in person. So the moment we can actually do this live, I'd be there as a shot. So it was a great honor to be invited, and I enjoyed it, and the questions were superb as well. So yeah, spot on. I'm glad you found it entertaining, hopefully enlightening anyway.
Lovely. Okay, there we are. Just again, just a few final notices. Our next NEMI lecture of this type will be in May by Stephanie Glendinning on geotechnical assets. And don't forget, we'll be sending around further information about our 2-day conference with the Durham Energy Institute, and we'll hopefully see you there. So I think back to you, Steve, just to close things off.
Just to finalise everything, well, thank you everyone for attending this evening's lecture, especially Martyn. I think he's given us a heck of a lot to think about there, and we can see a future, there's no doubt about it, with so many different things in the future. So I'm sure everyone will agree it's been a very, very enjoyable and informative evening. So we'll hope to see everyone at our next lecture, which Andrew has just mentioned. So there's nothing else, I think, for us to say other than good night, and thank you very much for attending.
Yes, well, if people want to hang around just for a bit longer, just if they fancy a conversation, but I think, David, if you just press the kill switch for YouTube.
I will go away and have my dinner.
Oh,
thank you very much.
No, can I ask you one last question? Because I remember seeing these materials down at the Materials Expo in London, and it was the smell, which just you can't get on a virtual meeting.
Yeah. Well, which ones did you smell, Andrew?
I can't quite remember, but it was
smelly ones.
Something brick shaped like that, and it had a very strong sort of yeasty smell to it.
Yeah, so there's the yeasty smell from the mycelium that you'll certainly get. Now that's more to do with the substrate than it is the mycelium itself. But grown on coffee grounds, you'll get a bit of a coffee smell as well quite often. But the real killer are the bricks. So after they've been cured and they've been kind of cleaned, they're okay, but they come out smelling of ammonia, because that's basically what's causing the pH change. And so you get my lab stinks when we have casting days. Bespoke Concrete don't know about this yet, I don't think, but that's one of the drawbacks of our method, is that at least in the initial stages, the materials are quite smelly. Yeah, absolutely. If I were with you, I'd have brought some samples.
Oh, that would have been excellent. So there we are. Lovely. Right. Okay. We shan't detain you from your dinner any longer.
I'm going to have my dinner. I'm going to have a beer, I think. All right. Thanks, guys.
Enjoy, Martyn.
See you soon. Bye.
I think we're still. Derek, was that all right? I think we're still on YouTube as well, David.
Yes, it was. Yes, it was fine. Yeah, it did very well.